Hi, everybody. Welcome to your last unit and the reproductive system. So today we're going to talk about the female and the male reproductive systems separately, but first I'll go through an overview for you guys. So our reproductive systems, like other terrestrial animals, humans have the ability to internally fertilize. That allows flagellated or sperm which have flagella, which basically just means the tail that you see here, um, which gives them the ability to move. It causes them to be able to remain in an aqueous environment while they're searching for the oocyte. Humans mate for pleasure as well as procreation, and mating as well as courtship has different types of rituals, as do other animals. Human females are always sexually receptive, which just means that they are always able to be reproductive, so fertile, after puberty, and not just during what you would call quote unquote fertile periods. There are some species that just go through seasons of being fertile. Those are seasonal breeders. Obviously, humans are not seasonal breeders. Females after the point of puberty are always sexually receptive. The embryonic and fetal development is also internal. So that means inside of the uterus. The uterus is going to be there for protection of the developing embryo or fetus from dehydration, and also protects as like a cushioning. Humans are sexually dimorphic, which means there's there's physical distinctions between a female and a male. There is a lot of psychological debate over dimorphism and behavior and the psychology between a male and a female. So that kind of branches into, do sex hormones play a significant role in developing human behaviors, as they seem to do in other mammals? So that's kind of a good example of that is, do girls like dolls and little boys like guns, or is that a cultural thing? Obviously, that's outside the scope of this class, but that would be addressed in a psychology class. So we'll talk about sex determination to begin with. Female and male sex organs are going to consist of three different structures. So gonads, internal genitalia, and external genitalia. Gonads are going to be the organs that produce gametes. So for males, males will produce sperm, and females will produce oocytes. Sperm comes from the testes, and the oocyte comes from the ovary. Internal genitalia would be accessory glands, ducts, um, that take the gonads into the external environment. And then external genitalia is going to be all of the external reproductive structures. Sexual development is going to be programmed within your human genome, as you see here. Every nucleated cell of the body, except for the germ cell, is going to contain 46 chromosomes, which means we would call them diploid. So there is 22 pairs, as you can see here, of autosomes. Autosomes are going to provide the individual with different types of characteristics. So it will, these chromosomes will help code for things like their eye color, their hair color, the type of blood they have, if they have freckles or not, their height, all that kind of stuff. There is, however, one pair of sex chromosomes that are going to be designated to determine whether this individual is a male or a female. So X are going to be, X and Y are going to be the names of those chromosomes. And they contain the genes necessary for directing the development of both the internal and the external sex organs. The X chromosome is larger than the Y, as you guys can tell here on the bottom, which literally just means that the X chromosome has more genetic information on it in comparison to the little y right there. So if you have an XY, this would equal a male. If you had an XX, which would basically be two big chromosomes next to each other, that would identify a female. The germ cells, so the sperm and the oocyte, are going to be haploid chromosomes, and when they unite, they join together, 23 plus 23. So haploid is 23, excuse me. When you add 23 plus 23, you get 46. 46 would be considered the zygote. Zygotes are the fertilized cells that contain the unique set of 46 chromosomes, which basically means you got half of the chromosomes from mom and half of the chromosomes from dad. Okay, so sex chromosomes are going to determine the genetic sex. So the sex chromosomes of the zygote determine the sex of the individual. Again, genetic females are going to be XX, as you see here, and genetic males are going to be XY, as you see here. So in society today, just a lot of people say like, oh, I really want a boy, or oh, I really want a girl. The interesting part about that is if you are a female and you really want a boy or a girl, it doesn't really matter what you want. What really matters is not necessarily what dad wants, but what dad gives. So if you guys notice here, the female who has XX, because that's the designation for female, is always going to give her offspring the X. So whether it's the female or whether it's the male, she gave, right, the mom gave her offspring or kid the X chromosome. The male parent has both one Y and one X. Flip that, right? One X, one Y. Which means that he can either give his offspring the X and make the offspring a female, or he can give that offspring the Y and make the offspring a male. So it doesn't necessarily matter what you want. If you're a female, you have no control over it whatsoever. Obviously, if it's a natural birth, you can absolutely play with genetics inside a lab and pick what you want, but that's also not ethical. That's a different class, right? That would be bioethics. So XX is female, XY is male, and the male is always going to determine what the offspring is, either a boy or a girl. So in genetics, if we jump back to this, it's called a karyotype. These are chromosome pairs. So you've got one chromosome pair here, two chromosome pairs, 3, 4, and this again is called a karyotype. What happens when you do something different and you add in like a, let's do this, a third, we'll call it pair, right, to the group? It becomes a little bit catastrophic or a little bit different. In some cases, you would call it a mutation. So genetically speaking, having the presence or absence of extra chromosomes is fatal unless it's only one extra or one less on specific chromosomes. So you can have one extra or one less on chromosome set 23, 21, 18, and 13. So these you can have one extra chromosome on, and then the sex chromosome you can either have one more or one less and still make it to birth. So if, for example, you added on a third chromosome to set number 1, this offspring would always cause a fatal mutation and the embryo would die in utero, which means during development, it would end in what you would call a miscarriage. We, however, are talking about the sex chromosomes. So we're going to go back into talking about if you're adding one extra chromosome to the sex chromosome group, which is 23. So the presence or the absence of the Y chromosome will determine if the development proceeds either as male or female. There are certain exceptions, or not exceptions, but there are certain mutations that we'll talk about. So this is the first one. It's called Klinefelter syndrome. This is where an individual ends up getting on accident the X from mom and then also an extra X and the Y from dad. So the Y from dad is going to determine that this individual is going to be a male. But because he has the extra X chromosome, this male will sometimes have cognitive impairment, will have more female characteristics like more breast tissue, will have less musculature, is more likely to get female diseases like breast cancer. He will be infertile or have very reduced fertility. You guys will follow here on the side, right, where it says he can still be tall in stature, but his shoulders are going to be narrow, just because typically females don't have very broad shoulders, smaller testes, and infertility. The next abnormality we'll talk about is Turner syndrome. Turner syndrome, in regards to genetics, it looks like this. You have an X chromosome, and then you kind of just put a 0 or an O, which is basically just a placeholder. Really what happened is this individual only has one X chromosome. So with only one X chromosome, the zygote will develop into a female because there is a lack of a Y, but the individual will not have normal reproductive function. So common signs and symptoms of Turner syndrome would be infertility, short in stature, have a webbed neck, which is kind of the thickening of the neck, cognitive defects, and then sometimes we'll have congenital heart diseases. The last one that we'll talk about, okay, well, there's no picture, is the YO, and that's going to be a zygote with the Y chromosome. But because, and this makes sense, there's no picture here because with only one Y chromosome, you're missing the X chromosome. And if we jump back to that karyotype, you guys, we just talked about it. The X chromosome carries a lot of really important genetic, we'll just call them patterns, or it codes for a lot of different traits. So you cannot live without the X chromosome. So the YO will not survive without the X chromosome, which means that individual would die in utero. In a normal XX female development, once the ovaries have fully developed, one of the X chromosomes in each cell will actually inactivate, and they'll condense into what we call a bar body, which leaves only one active X chromosome in each adult cell. So we'll talk about that a little bit more next time when we go into meiosis a little bit more in detail, but that is important because really females or males, you only have one active X chromosome in the adult cells. Okay, so sexual differentiation and how it occurs in early development. So the sex of an embryo is difficult to determine early on because the reproductive structures don't begin to differentiate until about the 17th week. I'm sorry, the 7th week. But still you can't tell a ton from the outside. So that takes 7 weeks is about 40 to 50 days after conception. The embryonic tissues are considered bipotential at that point, which means that they cannot be morphologically identified as either male or female, and are considered indifferent in regards to their gonads, just because they haven't developed enough on the inside or the outside to have a doctor differentiate between the male or female just yet. The bipotential embryonic ducts that will form internal genitalia are going to be called Wolffian or mesonephric ducts, and then the Mullerian ducts as well. As development progresses, one pair of the ducts will develop and the other one will regress. So I'm gonna make these pictures just a little bit bigger for you guys. You guys can see this is week 6 for the fetus. This is the bipotential stage where you see the Wolffian duct, which is the yellow kind of comb looking thing, and then the Mullerian duct, which is it kind of looks like the optic nerve on the outer side. Gonads are inside and they're still bipotential at this point. Nothing has been determined just yet. The bipotential external genitalia will consist of a genital tubercle, urethral folds, urethral groove, a labiosacral swelling that will eventually differentiate into either the female or the male reproductive structures as the development proceeds. These are the external genitalia. The sex determination, therefore, then is going to depend upon the presence or the absence of what you would call an SRY gene. So the SRY gene is the sex determining region of the Y chromosome. So the functioning SRY gene will result in male testes, while the absence of the gene will cause the gonads to develop into ovaries, which would make the individual female. So the SRY gene will direct male development. Again, if it gets turned on, right, it's going to start producing testes determining the proteins. It will initiate the production of the gonads in the testes, which will then cause the production of Leydig cells and Sertoli cells. Leydig cells will secrete testosterone, which will help the development of the male reproductive structures, while the Sertoli cells will secrete what's called an anti-Mullerian hormone, which causes the regression of the Mullerian duct and then will allow for the further the male reproductive structures to continue to develop. Okay, so the male and female embryonic development will proceed kind of as follows. So you guys can see here you have the bipotential up here at week 6, and then on the left you're going to follow the female path, and then on the right you'll follow the male path for the structures that are going to develop. So during the first 40 to 50 days of embryonic development, the reproductive structures are indifferent and have bipotential. So we're up top. If a Y chromosome is present in the embryonic cell, the SRY gene will produce a protein called the testes determining factor, or the TDF. The female embryos do not have the SRY gene. So that's kind of step 1, right? The SRY protein in a male embryo is going to start to direct the medulla. Oops, sorry, I got a little. The medulla of the bipotential gonad to develop into the testes. The TDF will bind to the DNA and activate additional genes that are going to direct gonadal medulla to form the testes. The absence of the protein signals are going to trigger the cortex from the ovarian tissue, while the medulla will then regress. So on the female side, the gonadal cortex becomes the ovary in the absence of the SRY protein and under the influence of female-specific genes. Once the testes is formed in the male, they begin to secrete 3 different hormones that are going to influence the development of both their internal and external genitalia. Again, one of them is the Sertoli cells. The Sertoli cells will secrete the anti-Mullerian hormone, or it's also called the Mullerian inhibition factor. Either way, it's saying that you're suppressing the Mullerian. The Leydig cells, again, will secrete testosterone and therefore DHT, which is a dihydrotestosterone that will bind to the same receptors, but it will cause different responses to occur. With the Mullerian, so the MIF, which is the Mullerian inhibition factor, it causes the Mullerian ducts to regress, while testosterone stimulates the Wolffian duct to form the male structures like the epididymis, the vas deferens, and the seminal vesicles. So the Mullerian duct was here. You guys can see it in the dotted line. Again, because of the MIF, the Mullerian duct is going to regress. And what's going to happen is the Wolffian duct, right? So the Wolffian duct was the inside one. The Wolffian duct is now going to start to develop things like the epididymis, the vas deferens, and the seminal vesicle. The DHT. So the DHT is going to control the differentiation of other male sex characteristics like the external genitalia and the prostate gland. For females, so jump back to the left, the absence of MIF, in the absence of MIF, your Mullerian duct is going to develop. So the Mullerian duct will develop, and it's going to start to form the upper portion of the vagina, the uterus, and the fallopian tube, while the Wolffian duct will now regress or degenerate, and it will not influence testosterone. So now you can see that the inside tube is going to regress, whereas the outside one is going to develop into the uterus, the vagina, and the fallopian tube. So without the DHT, the bipotential external genitalia takes on female characteristics. There are disorders of embryonic sexual development, so like hermaphrodites. So hermaphrodites would be a condition in which both the ovarian and testicular tissue are going to be present in the body. 34% of hermaphrodites have an ovary on one side and a testes on the other, whereas 20% have what we would call ovotestes, which means part of the testes and part of the ova on both sides. 46% would have ovotestes on one side and an ovary or a testes on the other side. So these conditions are extremely rare and typically result when some embryonic cells receive a short arm of the Y chromosome with the SRY gene, whereas most of them do not. Another disorder would be a pseudohermaphrodite. The pseudohermaphrodite is more common, although still pretty rare, where you have an individual with either the testes or ovaries, not both, and they have accessory sex organs and external genitalia that are incompletely developed or inappropriate for their chromosomal sex. The most common cause of the female pseudohermaphrodite is a congenital adrenal hyperplasia, which is a condition that can be inherited by a recessive trait and cause the adrenal cortex to secrete way too many androgens. Since the cortex does not secrete the MIF, the female with this condition will have a uterus and fallopian tubes, but also the Wolffian duct derivatives and partially masculinized external genitalia. In males, one of the interesting causes of pseudohermaphroditism is testicular feminizing syndrome. These are when you have males with this condition, they have normal functioning testes, but they lack the receptors in the testosterone. Embryonic tissue would be unable to respond to testosterone, and therefore female external genitalia would develop. In this case, because of the MIF, the production by the testes, no internal female structures would develop. A child appears female in this case, but is chromosomally male and has no accessory organs. Other male pseudohermaphrodites have normal functioning testes, normal testosterone receptors, but they genetically lack an enzyme. It's called the 5-alpha reductase, which is needed to make the DHT. The 5-alpha reductase deficiency would result then in male accessory organs internally because of the testosterone, but externally would be female characteristics because of the lack of the DHT. Without talking about these conditions, if you're talking about the regular development of male and female genitalia, somewhere around the 5-week point, the 10-week point, you're going to start to develop the external genitalia, which again would be female. In the absence of the androgens, the external genitalia would become feminized. If you're talking about the male, the DHT causes the male development externally, and the testes would descend from the abdominal cavity eventually. So this is it's called gametogenesis, which is the making of gametes. This is something that we'll talk about next time a little bit more in depth, but basically what happens is you go through a lot of cell division, then you get to the point where you need to have sexual development, which is meiosis. You're making gametes in this case. You're going to go through 2 different divisions, and you're going to end up with either oocytes from the female or sperm from the male. And again, you guys, we're going to cover this a lot more in detail, so I'm just going to kind of leave it there for today. Hormonal control of reproduction is something that we talked a little bit about already in the endocrine system. So in both sexes, the brain's going to control reproduction through the GnRH, which is the gonadotropin releasing hormone, as well as the pituitary gonadotropins like the follicle stimulating hormone and the luteinizing hormone. So if you guys will just review those really quick, because they do help with the production of the reproductive system and glands and such, but we've covered those extensively already. This basically takes you into the anatomical side of the male reproductive system. A lot of this will be in the lab portion that we will cover or have covered. But the review for the lecture side of it is you'll want to remember the components here and their function as well as the source. So for males, you'll need to remember where sperm is made, which hopefully you remember is in the testes, specifically in what we call the seminiferous tubules. Then you'll need to know the route of sperm throughout the body. So let's cover that real quick. Route of sperm, it's the testes make sperm. The sperm mature in the epididymis. When we say mature, hopefully you guys remember that would be the head of the sperm, which is made in the testes. In the epididymis, they get the flagella, which means the tail, and it's for the function there is for movement. So testes, epididymis, then they go through the vas deferens or the ductus deferens. Then they go through the seminal vesicle, prostate gland, and then maybe not through is the best word, but they gain the secretions from the seminal vesicle, the prostate gland, the bulbourethral, which is also called Cowper's, and then they go into the urethra and the sperm would then come out. So there's different points here. You would call this like the prostatic urethra and then the penile urethra and then all the way out for the sperm. The really important parts again is that sperm have a lot of barriers that they have to overcome in regards to fertilizing an oocyte, which would basically mean creating a zygote or an offspring or a baby. The female reproductive system is very acidic, and because it's very acidic, it means that a lot of the sperm are going to be killed off like instantly upon entry. But the sperm have their own way to kind of combat that. So if you guys are looking here at the head of the sperm, this is called an acrosomal cap. The acrosomal cap has enzymes that are going to help do two things. So one is the enzymes are going to help break down the barrier of the oocyte so that the sperm can fuse with the oocyte. And then the second thing is that the acrosomal cap will release a alkaline fluid, which is going to buffer out the acidity of the female reproductive system. So you'll want to go through and talk about the bulbourethral glands that are for lubricant, which is mucus, which basically just means that it's going to help the movement of the sperm so they don't get stuck. The bulbourethral gland is going to, again, help neutralize the acid from the vagina as well as the prostate. And then the prostate will also contain the enzymes. Lastly, you'll talk about the seminal vesicles as giving the nourishment to the sperm. They provide fructose, which is the energy for the sperm to move. This would just be a review. This would be straight lab. And that takes us into female reproductive system. So this is your absolute review. You guys definitely need to know that kind of stuff before we get into the female reproductive system. So female reproductive system has a little bit more to it than the male reproductive system. This is the anatomical side to it. So you'll hopefully remember, right? This is the uterus. This is the vagina. These are the ovaries here and here. I'm not sure if you guys are aware of this or not, but every month, only one oocyte gets released from one side of the body. So obviously you've got an ovary on the left and the right if you're a female. One oocyte gets released into the fallopian tube and then will go into the uterus each month. So every month the side alternates. Some people are in tune enough with their body to know what side, just dependent upon the cramping that they feel, and would be able to tell you, this is the side that I'm ovulating from this month. Others can't. So let's get into, so this is all the anatomy, which we've covered already in lab, but let's get into the development of the follicle. So when we talk about the development of the follicle, we can do so in a chart like this, which is very good because it's everything stacked on top of each other, and it's going to show you what happens with your gonadotropic hormones in relation to the ovarian cycle, so the development of the follicle, as well as the ovarian hormones, as well as the uterine cycle, which has to do with the lining of the uterus. It will also break it down for you in regards to the phases that the menstrual cycle goes through, as well as the body temperature throughout each one of those phases. So if I back out for you guys just 1 second and go back into the ovary has what we would call follicles develop to the point where they're mature and would be able to be expelled out of this ovary. The only way that the oocyte, when fully mature, can be expelled is with the presence of the luteinizing hormone. The luteinizing hormone is going to spike and it's going to allow ovulation to take place, which means the oocyte is being released out or propelled out of the ovary and then will be captured and put into the fallopian tube. So that will be the first thing we'll talk about. So this menstrual cycle should be divided into about 28 days. And the gonadotropic hormones will peak, both of them, FSH and LH, just before ovulation. Now, obviously, the spike for LH is much higher than it is for FSH because the spike for LH means that the luteinizing hormone is helping, again, the release of the mature oocyte out of the ovary. FSH is the follicle stimulating hormone, and it's actually used all the way throughout the 28-day cycle. So you develop from primary into secondary, secondary into tertiary. Now tertiary, they're ready to be expelled. And then you have the regression and then the redevelopment on the other side of it. So FSH is used all the way through. It does peak a tiny bit just before ovulation because you're preparing for the mature follicle to be released. When you correlate that to the ovarian cycle, the first day of the menstrual cycle is going to be a primary follicle. So this is when the follicle is just beginning to develop. At ovulation, you have the expulsion of the mature follicle out of the ovary. So that means the mature follicle has to be developed before ovulation. So in the late follicular phase, the tertiary or the Graafian follicle is developed. Ovulation, again, is obviously the process of releasing that oocyte from the ovary. And then you have the corpus luteum form, then mature, and then corpus albicans. And then, so this is the regression of like that area. And then what basically is happening is your body is preparing. So it's regressing, getting rid of anything that's left over, and then preparing to make a whole new follicle the next time. The ovarian hormones, so you have estrogen, inhibin, and progesterone. Estrogen is going to peak very, very, very highly just before ovulation. That will help as well with the body getting ready for the oocyte to come out and potentially be fertilized. Inhibin is also going to peak just a little bit before ovulation. And then progesterone is actually one that is going to peak way after ovulation. So it's going to occur in what we'll call the luteal phase. So the last portion of the menstrual cycle. The progesterone spike is actually going to be correlated to the uterine lining maturing or developing or thickening. So if we jump down to the uterine lining, what happens in regards to the days of the cycle? So days about 1 through 5 or 1 through 7 is going to be what we would call menses. This is also going to be accompanied by bleeding. It's basically the shedding off of the endometrial lining of the uterus. It's because there was nothing nothing meaning there was no fertilized egg that took place and embedded in the endometrial lining, which means that the body is like, okay, I'm not pregnant. Let's get rid of this inside layer and let's build a new layer to make sure that it's as functional as it possibly can be, because that layer that we're talking about in the uterus, again, it's called the endometrial lining, would be the layer that's going to house the fertilized egg, which means it's a zygote, which means this layer is providing the nutrients and the protection and everything that this zygote needs to become a baby. So what happens in the first part, it's menses. It's basically you're getting rid of the endometrial lining. Then you're going to go into what you'll call the proliferative phase. So if something is proliferating, what that means is it's growing. So what's growing in this case is the endometrial lining. The endometrial lining gets built back up because, again, it has to be fully ready to take on a fertilized egg and be able to house it and nourish it if it was to be released and embed in the endometrial lining. At days 14 through 28, the only thing that's going on in the endometrial lining, which is the uterine cycle, is the continual development of that inside lining until it's as functional as it can be, so it's ready to house that fertilized egg. Body temperature. So what happens with body temperature is it's normal, it drops just a tiny bit. Excuse me. And then it peaks just after ovulation. So some people will do things like take their temperature to try to figure out when they're ovulating, when they see a huge spike in their body temperature. And by huge spike, I mean 2 degrees. 2 degrees Celsius, excuse me. So when they see a spike in their body temperature, they'll be able to definitively say that they should be ovulating, which some people use as kind of like calendar counting to either try to or try not to have a baby. This is everything that we just talked about in regards to the ovarian phase and the development of the follicle. These are the hormonal changes that we just covered for the menstrual cycle of LH and FSH, excuse me. Fertilization. So fertilization is where you have a sperm and an oocyte fused together. So fertilization must occur within 24 hours of ovulation. This picture shows the difference in the size of a sperm and an egg. Obviously, the egg is much, much larger than the sperm. Really, what happens is there are tons of sperm that are trying to fertilize that one egg. So you would say a capacitated sperm is going to release enzymes from their acrosomal cap in order to help penetrate or fuse with the oocyte or the layer of the surrounding egg. The first sperm to fuse with the egg. So here's a really good question. If there's so many sperm trying to fertilize this one egg, why do multiple sperm not fertilize it? So what happens is the first sperm to fertilize the egg fuses with it and it releases its nucleus. The nucleus then of the sperm will fertilize with that of the egg. While all that is happening, the sperm is also putting off this chemical signal. And it's basically like, I got here first. You can't touch the egg. So basically, what happens is the first sperm to fuse secretes this signal that's like, you can't touch in regards to all the other sperm. So none of the other sperm will be able to fuse with this egg because that other sperm got there first. Timing of ovulation. So ovulation is the expulsion of the oocyte. Then it takes a couple of days for the oocyte to move. So what happens is the egg or the oocyte gets swept into the fallopian tube. Fertilization should take place somewhere around day 1, right? Day 1 to day 2, somewhere in there. So if you guys notice, obviously the little swimmers, right? Those little sperm made it all the way up here, but reality is they had to start way down here in the vagina. They had to make their way up. And then I don't know if you guys, are aware of this or not. Here, let's go to this picture. Really what happens is, so we'll call this area here like the vagina, the sperm come up and around, and then half of them are going to go this way. The other half approximately are going to go to the right. This example is showing you that this ovary released this oocyte this month. So this is the only half that can actually fertilize something. These half, they're traveling and they're going as fast as they can, but there's nothing to fertilize once they get there. So there's a lot of barriers that the sperm have to overcome. They have to go a literal long distance. They have to go uphill, which means against gravity. They have to encounter the acidic environment of the female reproductive system. They have to choose the right direction. They have to be very fast and get there first. And then when they do so, they have to properly fuse with the egg. So if all that happens and takes place, what happens there is fertilization occurred, the zygote would form, and then the zygote would go through development and divisions. So day 2 to 4, the cells divide. Day 4 to 5, now you'll call this a blastocyte, and it's now going to reach the uterus. Somewhere between day 5 and day 9, the blastocyst is going to actually implant in the endometrial lining of the uterus and then begin to develop from there. When that happens, a placenta is also developed. So the developing embryo floats in amniotic fluid. It obtains oxygen and nutrients from the mom through the placenta and the umbilical cord. So the placenta is all around. The umbilical cord will provide the nutrients to the fetus. And obviously, they don't have fully functional heart or lungs just yet. So the mom will produce everything that the baby needs, which is one of the reasons why pregnant mothers are so tired, because they're providing tons and tons and tons of extra energy into the development of this fetus. Okay, so parturition is basically birth. So the birthing process, I'll go into this a little bit more next time as well, but basically it's broken down into a couple different stages. The first one is the fact that the fetus has to be fully developed. So as labor begins, the fetus should be head down in the uterus, and the head will push on the cervix and dilate it or make it bigger. Cervical dilation will cause the cervix dilating will cause neurons to fire to the brain and then tell the pituitary gland release the oxytocin. The oxytocin then will target the uterus and specifically the myometrium, which is the muscular layer, and it will cause contractions to occur. The uterine contractions push the head against the cervix more and causes more stretching and more dilating until the mom is fully dilated, which is 10 centimeters, and the uterine contractions will then help the fetus be pushed through the vagina. That's obviously a natural birth, at which point the oxytocin can be cut off and now can be rerouted back to the mammary glands so that the infant can now also get milk from the mom. Milk from the mom comes from the mammary glands. This is known as lactation. The mammary glands are modified sweat glands that are going to secrete milk into the ducts of the glands and provide the infant for all of the nutrients that they need. The first couple days, the mammary glands are not actually producing milk. They produce it's like a thick white solution, which has a lot of fat in it. And the fat is just to kind of bulk up the baby for the outside, after which point mom's milk will come in and then milk will be provided until the baby stops and is weaned off, and the milk will, or the glands will stop producing, because the body will know, I don't need to provide anymore because the baby's not eating anymore, which means I can stop making the milk. So that's all making a baby. So how to not make a baby. Contraceptive methods and then efficacy. So how efficient are they? So no contraceptive typically will result in about 85% pregnancy. These numbers are from 2013, so they're almost 10 years old, which means that these numbers aren't exactly accurate anymore. I think this number has gone down a little bit, and I think there's been a little bit of an increase in infertility in the last couple of years. But continuing on with contraceptives, spermicides. So it literally is what it sounds like, right? If you're thinking like a pesticide, pesticides kill bugs or pests. A spermicide is going to kill the sperm, and that results in pregnancy about 30% of the time. Abstinence during times of predicted fertility. So that would be like counting on a calendar, like, oh, I'm ovulating and I shouldn't try to get pregnant during this time. So that works about 75% of the time. Diaphragm, cervical caps, or sponges work somewhere between 16 and 32% of the time. Oral contraceptives, flip it. They work, except for during that time. Oral contraceptives work about 92% of the time. IUDs about 99. Contraceptive hormone injection 99. Condoms about 85. Female condoms about 79, and sterilization over 99. So that would be a vasectomy for a male or tube tying for a female. We'll cover a little bit more of this in development, and we'll end our semester there. If you guys have any questions, let me know. I'd be happy to answer them for you either in the discussion boards or in email. And I will see you guys back one last time for our last lecture. Have a good day.